Hi Platypus! Welcome to Platinum, where future top-notchers are made. I am Shane, and join me as we make our journey to Top 1. So what comes to your mind when you hear the word cholesterol? Most people think of something bad, a risk factor for heart disease, or something to avoid in food. But here's the twist. While cholesterol does carry a risk in your bloodstream inside your cells, it's a lifesaver. It helps your cell membrane stay stable and functional, especially when things go south. Today's discussion centers on the role of cholesterol in membrane fluidity, and our learning objectives for this session are as follows. At the end of this session, the students should be able to first, define membrane fluidity and explain its biological significance using the fluid mosaic model. Second, describe the structure of cholesterol and its orientation within the phospholipid bilayer. And lastly, discuss the cholesterol's buffering effect and how it stabilizes membrane structure during environmental fluctuations. Hopefully, by the time we're done, you'll see cholesterol in a whole new light. Let's start with the basics. Remember the basic layout of the phospholipid bilayer. So each phospholipid is an amphiphilic molecule, meaning it has a hydrophilic head that loves water and two hydrophobic tails that are scared of water. Because of this, when placed in an aqueous environment, these molecules self-assemble into two layers, a bilayer. In this arrangement, the water-attracting heads orient themselves outwards, facing the surrounding water, while the water-repelling tails are sequestered inward, forming the core of the membrane. If the heads love water and the tails hate it, what would happen if a phospholipid tried to flip itself upside down in the membrane? Wouldn't that work easily? If this were to happen, the cell membrane would become highly unstable. Now, imagine this boundary isn't a stiff wall, but a super active surface where these phospholipids are constantly wiggling, spinning, and sliding past each other. This constant motion is called membrane fluidity. It's all about how freely molecules move within the lipid bilayer of a cell membrane. To make sense of this, scientists proposed the fluid mosaic model. The fluid mosaic model depicts the cell membrane as a dynamic, mixed structure. Why fluid mosaic? Fluid because its lipid and protein components are constantly moving, largely due to the flexible phospholipid bilayer. Mosaic because of the diverse embedded molecules within this moving lipid environment. Proteins for various functions, carbohydrate chains for cell recognition, and our special guest, cholesterol for membrane fluidity regulation, all present together like a picture made of many different tiles. Now, what does the word fluid suggest about the nature of the cell membrane? Exactly. This fluidity allows cells to deform and migrate to facilitate membrane fusion for transport of substances into and out of the cell, and to enable the interaction of membrane proteins for cell signaling and enzymatic activities. But remember, this fluidity isn't fixed. It's strongly affected by temperature and lipid composition. For instance, colder temperatures tend to make the membrane more rigid, while warmer temperatures increase fluidity. Similarly, the types of lipid present, particularly the saturation of their fatty acid tails, significantly impact how tightly they pack together. Saturated fatty acid tails are straight and can pack very closely, leading to a more rigid membrane, while unsaturated fatty acid tails have kinks that prevent tight packing, promoting greater fluidity. Now, let's see how cholesterol fits into this picture. Similar to the phospholipids in the cell membrane, cholesterol possesses both a hydrophilic or a water-attracting region and a hydrophobic or a water-repelling region. It has a small polar hydroxyl group that interacts favorably with water, a big, rigid steroid body that repels water, and a short hydrocarbon tail that also repels water. Why is it important that cholesterol has both hydrophilic and hydrophobic regions when inserted into the membrane? This specific orientation sets the stage for cholesterol's powerful role in buffering membrane fluidity. Because of its mixed nature, cholesterol cleverly squeezes into the phospholipid bilayer. Its water-loving hydroxyl group snuggles near the water-loving heads of the phospholipids. Its rigid steroid body and hydrophobic tail then bury themselves deep among the phospholipid tails. This special placement is key to how it works. Think of cholesterol as the membrane's personal thermostat, keeping it just right no matter the outside temperature. When temperatures get warm, phospholipids get super active and spread out too much. This can make the membrane too loose and leaky. This effect is especially evident when the membrane contains many unsaturated fatty acid tails, which again have kinks that prevent tight packing. But cholesterol steps in. Its stiff body acts like tiny anchors sitting between the phospholipids and holding them closer together. It prevents them from spreading too far, keeping the membrane from getting too fluffy. What about it when it gets cold? In cold temperatures, phospholipid tails want to pack tightly, making the membrane stiff and brittle. This tendency is particularly strong with saturated fatty acid tails, which are straight and can align very closely. A rigid membrane compromises cellular movement and proper function. But again, cholesterol is there. By wedging its rigid body between the tightly packed phospholipid tails, it actually stops them from packing too tightly. It creates little gaps, keeping the membrane flexible and preventing it from becoming brittle. Now, question. Why is a floppy or leaky or too stiff and brittle membrane generally bad news for a cell? Consider a person experiencing a fever, their body temperature rises, or an athlete engaged in intense physical activity, localized tissue temperatures might increase. Conversely, think about exposure to a cold environment. In all these scenarios, cells must continue to absorb nutrients, expel waste, and communicate effectively. Without the cholesterol's buffering action, the cell membranes could become either too permeable or too rigid. As a result, these vital processes are disrupted. Clearly, cholesterol helps maintain the needed balance, especially in warm-blooded animals like us who must keep cell function constant despite environmental changes. Now, to bring our discussion to a close, let's quickly summarize what we've learned. We now understand that, dear, cholesterol is an essential membrane component. Its amphipathic structure allows it to embed in the bilayer, and its rigid core modulates lipid movement. At high temperatures, it stabilizes the membrane. At low temperatures, it prevents tight packing, keeping the membrane flexible but intact. This buffering action protects your cells across varying conditions, making cholesterol much more than just a dietary villain. And that's it. Thank you so much for learning with me today. And remember, strive, excel, succeed. This is Platinum, where future top-notchers are made. See you in the next review.